Hello everyone, welcome to Get My Class. So let's solve this question. The question says the gene isolated from dash producing ADA is introduced into cells at dash. It could be a permanent cure. Option A, bone marrow cells, early embryonic stages. Option B, epithelial cells, early adult stage. Option B, bone marrow cells, late embryonic stage. And option D says bone marrow cells, death. Now let us understand the question. The question says gene isolated from dash producing ADA is introduced into the cells at dash. It could be permanent cure. So you concentrate on the keywords that says gene isolation, production of ADA, and permanent cure. So when we talk about isolation of the gene, okay, which is actually responsible for the production of ADA. So what is ADA? Adenosine deaminase. Okay, deaminase. It is a type of enzyme. So this ADA gene will code for enzyme that is ADA. And this enzyme is necessary for defense mechanism. Okay, it is necessary for defense mechanism. Now, this gene, which is coding for ADA enzyme, this can be introduced into cells. Okay, and they are talking about permanent cure. So the cells should be in the embryonic stage, and it should be early embryonic stage because that time only the embryo will be developing. And further, when that particular time, if we introduce any gene, it whatever cells will multiply then, or you can say undergo division, it will also divide, or it will also have copies of this gene. Okay, so that's how that particular stage will give rise to permanent cure. It can be permanent cure. Now for this, we want the gene, and to isolate the gene, we want a particular cell because genes are present in the DNA and the DNA is present within the cell. Okay, so the cells what we are talking about the cells which have the capacity to produce ADA, that is adenosine deaminase. And which are the cells which are involved in defense mechanism mainly? That is nothing but particular type of WBCs. And where are these WBCs produced? These are produced in the bone marrow. Okay, these are produced in the bone marrow. Okay, first they'll be produced as stem cells, and further they'll mature to different types of WBCs. Now, from bone marrow, when they isolate the stem cells, okay, and they isolate the stem cells, and those stem cells will contain ADA gene, and that gene will be taken up and introduced into the embryonic cells, okay, early embryonic cells. So that now these embryonic cells will be capable of producing ADA enzyme, and hence their defense mechanism will be fine. Otherwise, what will happen? It will lead to SCID, severe combined immunodeficiency disease, if this enzyme is not released. But since we are introducing the genes in the early embryonic stage, that can be a permanent cure for SCID. Okay, it is possible. Now let us see. Option A says bone marrow cells. Yes, that is correct. Early embryonic stage, that is also correct. So option A should be correct. Option B says epithelial cells. So we cannot, isolate ADA gene from epithelial cells because these are produced by WBCs. Hence it is okay. Option B says bone marrow cells. That is correct, but late embryonic stage because most of the cells would have already specialized. Okay, so they would have differentiated. So we want stem cells which occur at the early embryonic stage. Hence it is ruled out. So it is ruled out. Option B is bone marrow. That is correct, but at the death. So at the death time, then what is the need of the gene therapy, right? So what is the option? The correct option is option A. I hope you understood the expression. Thank you.